Otherwise, I wouldn't be able to have a link there. Boy, that was really bad. I wish you would have asked me before I talked about the unknown notes. Now, that's not recorded. So just let me state, if you're just coming in to view this now, I have put up the unknown notes, and you must put in your unknown notes your unknown number. And let me go ahead and go here. And you must put in here your test request will be up here. And then any questions you have for me can be put up here. And I will probably in the comments send you your results for when the test has grown. And when you request a test, you're not going to get the answers immediately. Like the gelatin takes 1 week to grow. That's why I started it last Tuesday, because that one takes a week. Most of them take only a few days, but some of them take like 5 days to grow. And you won't get the results for 5 days. So you have to figure out what you're doing, and you should be using dichotomous logic. That's why you did the gelatin test on Tuesday, the gram stain on Sunday, and the PR lactose today, meaning you're using dichotomous logic for doing your tests. And the first test, I'm telling you what you're going to be doing because I'm trying to reduce the number of emails. This project will have about 6 to 7 emails per student. And I decided that's just too many emails for each student for one project, because there's at least 30 students, maybe 35 students in this class. And to give 6 or 7 emails for one project with that many students, it's just too many. I'll be overwhelmed. And so what I did was I decided you're doing these 3 tests. They'll be very good, useful tests for you and for getting you started. And that will reduce the emails down to 3 or 4 emails per student. And that's something I can live with. Okay? Any question about any of that? So in your unknown notes, you should put the date, which actually that's added for you, so you don't need to put the date. But you should put your test request. You should put what your observations are, what your conclusions are, and then any test you want to do in the future. And you should be using dichotomous logic. All right, let's close that down and go on and talk about Lab 9. This is the last lab for the course, complete Lab 9, which I'm going to talk you through here. It's on the identification of gram-negative bacilli. Complete the case studies, which are in the worksheet, and complete the Lab 9 worksheet. There are some reading to do, chapters 5 and 10, just a few pages of each. And you'll be doing some video clips and some virtual laboratory exercises. So the primary learning objectives, be able to interpret the results for the following biochemical tests. We're going to be working with the IMVIC reactions, and IMVIC is an abbreviation for Indole, Methyl Red, Voges Prescure, and that's so difficult to say, most people just say VP, and then the Citrate test. We'll also be doing a carbohydrate fermentation in this lab, and I think we dealt with that before. The motility test, and then looking for whether the bacteria can produce hydrogen sulfide, hydrogen sulfide test production. Now, some of the tests are done in combination tests like triple sugar iron or SIM test, and we'll talk about these combination tests.
You're not screen sharing.
I'm sorry?
But you're not sharing your screen.
I'm not sharing the screen. Oh, thank you. You should have interrupted me earlier. All right, now I'm sharing So the triple sugar iron test is a test which looks at hydrogen sulfide production and carbohydrate fermentation. There's a little bit extra in that, but that's mainly what it is. The SIM test looks at hydrogen sulfide production, motility, and the Indole test. The growth on McConkey's, we ran into that before. It looks at gram negatives can grow on McConkey's. It's selective and differential. And you'll look at lactose fermentation. So that's one of the carbohydrate fermentation tests. And then HE or hectone enteric agar plates are similar to McConkey's. Anyways, you use these biochemical analysis to help you identify the bacteria. And in today, we'll be identifying bacteria that are gram negative. And we'll look at gram negative bacteria that cause human infections. Any question about any of that? Why is my scroll bar not working? All right. So we'll be specifically looking at two families. The first is the Bacteriaceae family, a family of bacteria that are small, gram-negative bacilli. Get that out of the way. They're facultative anaerobes. They grow at 35 degrees, although they grow rapidly at room temperature as well. This is a very large family, 53 genera. 24 of the genera of this family are known to cause human infections. They're common and they're found in clinical specimens. They're all glucose fermenting and oxidase negative. So those are some of the characteristics of the members of the Bacteriaceae. There's a large number that cause human disease, and I'm sure you've heard of especially the first four: Salmonella, Shigella, Proteus, and Escherichia, like that's E. coli, Citrobacter, Enterobacter, Serratio, Klebsiella. You may have been less familiar of those, although you probably heard of Klebsiella pneumoniae. Morganella, Yersinia, Edwardsella, and Providentia. You may not have heard of those four. Most of these genera are part of your normal microbiota in the intestinal tract, and we call these, if they grow in the intestines, coliforms. But they can be causing opportunistic pathogens, meaning normally where they're growing where they should be, they are part of your normal microbiota, but if they grow in other places, or they grow in numbers that they shouldn't, they can be an opportunistic pathogen. Common bacteria that can be opportunistic bacteria, meaning the common genera that cause opportunistic infections include E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera are associated with human diseases such as gastroenteritis. That's where your stomach is upset, you get diarrhea or constipation. Can result from consuming fecally contaminated food products or water or drink, or improperly prepared foods where fecal to oral transmission occurs. And these include the genera Salmonella, Shigella, and certain strains of E. coli, like E. coli serovar 0157H7, which is a human pathogen. Any questions about any of that? All right. The other family that we're going to study today is the family of Pseudomonaceae, and Pseudomonas Genosa is the human pathogen, or you could also call it an opportunistic pathogen. The members of this family are similar to the family of Enterobacteriaceae, meaning they're all gram-negative bacilli. However, the Pseudomonas do not ferment glucose, so that's different from the Bacteriaceae, and they're all oxidase positive. So that's different from the Bacteriaceae. And so that's how you can identify the Pseudomonas as being different from the Bacteriaceae. Any questions about that? Okay, so some of the Pseudomonas are opportunistic pathogen, and with Pseudomonas aeruginosa, it's debatable whether it's an opportunistic pathogen or whether it's a frank human pathogen. It rarely causes disease in a healthy individual, but if the person has impaired, like a burn patient or immunosuppressed patient, it can easily cause disease in those patients. Pseudomonas aeruginosa is also implicated in healthcare-acquired infections, also called HAI, or nosocomial infections, and it is prevalent in the hospital. It's in the drains, the sinks, the water, in the water of flowers, even in dilute soap solutions. So Pseudomonas is found all over the place, essentially any environment where it's wet. It's important for microbiologists to be able to identify these species, gram-negative bacilli, and then culture them for clinical identification. And we use particular enzymes or byproducts to identify the different species. And here you're looking at a test. We're not going to talk about this test, but there's a test for identifying bacteria, and it runs a large number of tests at one time. And you're going to be looking at some biochemical tests to identify the bacteria. Now here's a question for you. Do you guys want me just to state the name of the test, or do you want me to briefly discuss each of the tests? Okay. I'm not hearing anything.
Briefly discuss them.
Okay, briefly discuss them. Anyone else? Because you can read it, but I can briefly discuss it. The Indole test is looking for the enzyme tryptophanase, which converts the amino acid tryptophan to indole and pyruvic acid. And you're looking at the production of indole, and if that is made, then the enzyme tryptophanase is found, and that's what the Indole test is looking at. You add Kovacs reagent to the tube, and if indole is present, you get a red color. E. coli test positive for indole production.
And if they're negative, do we just say, since on this chart it says the positive results and the negative results, do we put no change if it's a negative result? I know some of them do have a change as we get down below, but the top few didn't when I first started looking.
Okay, on this test, it's negative or positive. That's the conclusion. The result is it's kind of a pinkish color, and I know that looks rather purplish, but you can also call it reddish color. When you add Kovacs reagent, if indole is there, it'll be red, and it'll be yellow if you add the reagents, and it's negative, meaning no indole is present. Okay? Does that explain it? And
yeah, I looked at the oxidase on the virtual lab thing, and it just told me that it turned, the spots turned blue or purple, but it didn't say what the negative would be.
The negative is no color change.
Okay.
Okay.
Thank you.
Yeah. Sorry about that. Most of them, when they're negative, you add the reagent, and whatever the color of the reagent is, that's what color it'll be. With the oxidase test, the color is clear. And with, in this case, with when indole is present, the Kovacs reagent turns pinkish or reddish, and this one kind of looks purplish, and that's positive. Okay, each test, you will record it differently, and how you can take a look at that. Let me see if I still have that. I don't have it anymore. Let me go to my own result. Hopefully it's in here. You can take a look at the unknown chart example. Here we go. So some tests, the result, the conclusion would be plus or minus. And that looks like that'll be the case for gelatin, 6.5%, 7% sodium chloride, urease, nitrate. But look on the PR glucose, it's not negative or positive. It's negative. It can be A. Let me scroll down to show you that. There's an A and there's an A. And then it can be AG. Okay. And so you have to look up on the test what the possibilities are. Most of them are plus and minus, but some of them differ. And the V just means variable. Okay. So it looks like just the glucose ones, the PR test, are the only ones which are not plus or minus. No, there's hemolysis also is not plus or minus. Hemolysis, you have three. You can have negative, you can have alpha, or you could have beta hemolysis. Let's close that down. Come back to the test. All right, the next test you'll be looking at is the methyl red, and the test comes together, so it'll be the methyl red Voges test. And to do the methyl red, you just follow the directions for following methyl red. To do the Voges you just follow the Voges test. My point is it starts with the same tube. And if you were wanting to run both of them, you get two tubes. One you'd follow for the methyl red test, one you'd follow for the Voges test. All right, any question about that? For the methyl red test, you're looking at the mixed acid fermentation, where glucose, when it is fermented, it can produce acids. And if the acids are big enough to overcome the buffer in the methyl red test, when you add methyl red to the tube, it'll turn red, because methyl red in acidic conditions is a red color. And if it's not red, it will be yellow, is that right? Meaning if it's not acidic, methyl red, and you add it to the tube, it'll be yellow. Okay, so you're looking at the fermentation of mainly glucose, and if that glucose fermentation produces strong acids, you will be methyl red positive. And if it does not produce strong acids, meaning neutral products or some other products, then it'll be methyl red negative. Any question about that? All right, you'll also be looking at the Voges test, and it also is looking at the production of, meaning the glucose fermentation, but with the Voges test, we're not looking at acids produced. We're looking for the production of, here it is here, acetone, a neutral product of the fermentation of glucose. And if acetone is produced, the reagents will turn red after 30 to 60 minutes. And if acetone is not produced, then you'll be negative for a VP test, and the color will be yellowish. Okay? So both tests, the methyl red and the Voges are looking at glucose fermentation. With methyl red, you're producing strong acids. And if that happens, the methyl red will be red. And in the Voges test, you're looking for the neutral fermentation of glucose, and you'll produce acetone, which will be VP positive. It'll look red. So both of them, if they're positive, they look red, the methyl red and the Voges test. Any questions about that one? Or those two, I should say. Okay. Okay, in the sodium citrate test, the only carbohydrate the bacteria can utilize is citrate. And if the bacteria can utilize citrate, meaning metabolize it, it will grow on the tube, and the utilization of citrate will make the tube become alkaline, and it'll turn bluish in color, like this tube here. And that's because the bacteria can utilize citrate. You remember citrate is one of the carbohydrates in the Krebs cycle. So if the bacteria can undergo the Krebs cycle, it can utilize citrate. And if the bacteria cannot use citrate, it won't grow on the tube, and the tube will not change color. And we'll call that negative, and the tube will stay a green color. Okay? Once again, citrate is the only food source for the bacteria. And if it can't use citrate, it won't grow on the tube, and the tube will stay greenish in color. This tube, you won't have to worry too much about it because you'll be doing it virtually, but you can introduce a carbon source if you put too many bacteria on the tube, meaning you culture too many bacteria. So this tube should be slightly cultured, like with an inoculating needle, and you only put a little bit of bacteria on the tube, because the bacteria can utilize the carbon from a ton of bacteria if you put it on there. And an easy way to do it would be to take this tube, and if there's growth on it, streak the growth on this tube to another tube, and usually the second time you won't put on too much bacteria, and if it doesn't grow, then you know that you added too much bacteria to the first tube. Okay, any question about that? Both E. coli and Shigella don't metabolize citrate, but you can look on the chart there for that, for what species do. The carbohydrate metabolism test is also called the PR fermentation test. If the bacteria can hydrolyze a particular sugar and ferment it, then it will be positive for that test. And there's many different sugars you can use for this carbohydrate fermentation test, or the PR fermentation test. The ones that you can do are glucose, lactose, sucrose, maltose, and is it inulin? Okay, and there will only be that one sugar in the tube, and then you look to see if the bacteria can ferment the sugar. If they ferment the bacteria, the color will change from reddish to yellow, because the fermentation of the sugar produces acids, and the acid changes the PR, phenol red, to a yellow color. And if the PR is not acidic, it'll stay reddish. Okay? And it'll actually turn pinkish at the top if it becomes basic, but you don't need to worry about that part. Besides looking at the production of acids in the fermentation, you can also look at the fermentation producing a gas. And so there's three possible results in this test. You can get a negative, where the tube remains red and there's no gas production in the tube. The tube can be an acid, where the tube turns yellow, fermentation happened, but there's no gas production. And then you can have acid and gas where the tube turns yellow and there's a bubble in the inverted Durham tube. Any question about that? All right, so that's the carbohydrate fermentation test or utilization test, and the also called the PR, because phenol red is the indicator here, PR fermentation test. All right, the next test is the motility test. It's a tube, and it does have agar in it, but the agar is very low. And so if the bacteria is motile, it can swim in the agar and move away from your stab. If the bacteria is non-motile, which this tube is saying it's non-motile, then it will grow only in the stab. And I actually don't even see any growth in this tube, but there's a stab there because I can see the bubbles for where the stab is. Now this one is not very motile, but the stab is right there. You can see the thicker line right there. If the bacteria had remained only on the line, that would be non-motile. But it's moving away from the line, and you can see that it's extended from where the stab was. And sometimes if the bacteria is very motile, you'll see the bacteria everywhere. And you'll just look at the tube and you see that it's hazy everywhere. Okay, because the bacteria are growing everywhere. And that's how I'll show you the motility test. Another way to look and see if a bacteria is motile is you can just put the bacteria in a live mount, meaning a slide in water or saline, and then put a cover slip and then look at the bacteria under the microscope, and you can actually see the bacteria swimming around. Remember, bacteria, if they're motile, they have a flagella. Hydrogen sulfide production is looking at the ability of the bacteria to produce hydrogen sulfide. They're metabolizing sulfur if they're making hydrogen sulfide. And the bacteria that are doing this are usually engaging in anaerobic respiration. So you may only see hydrogen sulfide production at the bottom of the tube, which has less or no oxygen in it. And you might not see any hydrogen sulfide in the top of the tube because it has more oxygen. Usually hydrogen sulfide is looked at in triple sugar iron tube or a SIMS tube, the sulfide indole motility test. And both of these tests look at more than just hydrogen sulfide production, but that's one of the tests you can run from these two tests. You could also use the hectone enteric agar or the HE agar plates to look at the production of sulfide. And I'll show you that in a minute. Some bacteria that produce hydrogen sulfide include Citrobacter, Salmonella, and Proteus species. This is looking at triple sugar iron. I think maybe that's a SIMS test. Yeah, I think this is a SIMS test because that's going across. You stab the bacteria into the tube, and you notice how there's no black on the top of the tube. That's because hydrogen sulfide isn't being produced here. There's too much oxygen. But you see the entire bottom of the tube has hydrogen sulfide, and it turns the tube black. For the TSI tube, it's very similar to this. You stab the tube, and the hydrogen sulfide production will happen in the bottom of the tube. With the TSI tube, when you're inoculating it, besides stabbing, inoculating, you also grow bacteria on the slant of the tube. And what you can look at is on the tubes, hydrogen sulfide will look black, which is usually only seen in the bottom of the tube. The top is not black. And this is a complex test, but you can look at whether or not glucose is fermented and whether or not other sugars are fermented, lactose or sucrose, but you can't say specifically, although glucose can be fermented, but you can't say it's lactose and or sucrose, meaning it might be both lactose and sucrose, or it might be just lactose, or it might be just sucrose. You can't determine that. So on this test here, we can say that glucose is fermented because it turned yellow, and lactose and or sucrose are also fermented because it's yellow both in the butt and in the slant. If it's, wherever it's black, it's yellow. And so this tube is hydrogen sulfide production because it's black. And wherever it's black, I said it's yellow. So in the butt, it is yellow. And that means it is glucose fermented. Glucose in this tubes is at low concentration, very low, 10 times less than the other sugars. And so if glucose is fermented on the slant, the glucose will be used up. But on the butt, the growth of the bacteria is slowed down because it performs anaerobic respiration and it can perform aerobic respiration. You don't need to know about that. But the point is, if it's red on the slant and yellow on the butt, then only glucose is fermented. The glucose was fermented here and it initially turned red, but by the end of the test, all the glucose was used up. And then the bacteria start metabolizing the amino acids in the media, and that produces an alkaline substance. And so the color of the slant will change from yellow to red as the alkaline is produced. And so there we know, because it's red here and then yellow down here, that only glucose is fermented. The other sugars are at higher concentration. So if the other sugars are fermented, the slant will remain yellow. Is that clear? Anyways, this test is confusing to interpret, and you can look at this table here to interpret it. This one, no sugar is fermented because there's no yellow, no gas is produced, there's no bubble, and no hydrogen sulfide is produced because it's not black in the bottom. Okay, tube 2 I've already explained. Tube 3 is a tube where only glucose is fermented. That's why it's red on the slant, but yellow in the butt. The other sugars are not fermented. No gas is produced. There's no bubble. And then no hydrogen sulfide is produced. And this tube, it has a gas bubble, so gas is produced in the fermentation. Glucose and lactose or sucrose are fermented, and no hydrogen sulfide is produced because it's not black. All right, any questions about that?
Would the negative result for all of those be pink or red other than the hydrogen sulfide production? Did that start out as pink or red?
This tube is negative for triple sugar iron. No sugars are fermented, no gas is produced, no hydrogen sulfide is produced. Does that tell you tube 1? Okay.
Yeah.
All right. In the sulfur indole motility test, it's running 3 tests together. You're looking at indole production, you're looking at motility, and you're looking at hydrogen sulfide being produced in the tube. So let's go through and look at the control tube. Nothing is happening here. And this is probably a negative control, meaning it's not even inoculated. If hydrogen sulfide is produced, it's black in the bottom of the tube. If indole is produced, it will be pinkish or reddish when the Kovacs reagent is added, and that's why it's only pink on the top. The Kovacs is re-added, and that's a solid. If it's modal, you will see growth away from the stab, like this tube 4 shows motility, and tube 2, there's only growth in the stab, so tube 2 is not modal, and there's no hydrogen sulfide produced in any of these tubes, tube 3, 2, 3, 4, and 5. Indole is being produced in tubes 1, tubes 2, tubes 3, and tubes 4, because the Kovacs reagent has turned a pinkish color. And then tube 5 in the control, there is no indole, because it's staying a yellowish type of a color. And that's very light yellow. Okay, but you could call that clear.
How can you tell that motility is positive in tube 1?
It's not positive in tube 1. That's an, oh, excuse me, for that one. Because the black is everywhere, and the black will be where the bacteria is. But you're right, it's very hard to see for sure where the motility is because it's so dark. But because it's black everywhere, they're assuming the bacteria has moved everywhere, and it is motility positive. That one is just one that when we're looking at this, it's hard to see on this one because motility is everywhere. But if this was producing hydrogen sulfide, this region of the tube would be not black or very, very lightly colored, because there shouldn't be hydrogen sulfide production where there's no bacteria, okay? And you just sort of need to know, you can ask me a question about that. Like if your tube comes back like that, you say, you can ask me, is the motility everywhere on tube 1? It's all dark, and I'll tell you, okay? But that is modal. We'll just word it that way. All right, any questions about that? That is an excellent question, by the way. All right, you can use McConkey's and Hectone or HE, I'm not going to say it, it's just too difficult to say, to grow gram-negative bacteria. Both McConkey's and HE plates tend to select against gram-positives. And on the McConkey's, if the bacteria ferment the sugar mannitol, is that right? No, it's fermenting the sugar lactose. That's why it's LF, lactose fermentation. It will be pinkish in color. The colonies will look pink. And this one is producing so much acid that the pink is away from the growth. Everyone see that? If the bacteria on this side of the plate does not ferment the sugar lactose, it will not be pink. And the colonies here do not look pink. So you're looking at gram-negative, and then whether lactose is fermented or it's not fermented. You can do that also on the HE agar if they're fermenting lactose. What color do you say that is?
Yellow, red?
Yellow or orange, something like that. If it ferments lactose, it'll be that color. And if it doesn't ferment lactose, it will be a normal color, or this black color, meaning it's not orange, all right? And HE can also distinguish between the ability to produce hydrogen sulfide gas, and the colonies will be black if they produce hydrogen sulfide gas. And if they don't produce hydrogen sulfide gas, they'll be their normal color. Okay, any questions about HE and McConkey's? All right, that's it for the different tests you're going to be looking at. So in lab 9, view the clips on carbohydrate fermentation, on the SIMS test, and on the MR and the VP test, on the triple sugar iron, which is looking for the sugar fermentation as well as the production of hydrogen sulfide gas. You'll be looking at McConkey's auger, a video clip of that. And then also view some of the website Microbuzz. Click on the link differential tests. Once you get to Microbuzz, it's to the left. You only need to look at the tests that are needed to click to complete the table in step 1 of the worksheet for lab 9. And let me go ahead and show you that. So this is the table here. For some of these, whatever tests on the table, you'll need to get them from, where do I have it? Not there. Oh, here it is. You'll need to get it from Microbuzz. Any question about that? But there's a lot of tests on Microbuzz that are not in the worksheet. And you don't need to view them or learn about them. You only need to learn about the ones in this table here. You need to fill in this table. Okay, any questions about that? And then I guess I was wrong. There are no virtual lab for this one. You're just watching the videos and then looking at the Microbuzz webpage on the differential test. And then complete the case studies below. You're looking at a case study, which I'm not seeing the patient here. So anyways, there's case study 1, where a patient is infected and then they do tests to try and identify what the infection agent is and come up with what the infective agent is. Let me see if I can show you the worksheet. It's a little more meaningful on the worksheet. I already had the worksheet open. Answer the questions in the worksheet and do the case study, which you're using this table, which is given to you in the worksheet for identifying your unknown in the case study. What the heck? Where's the pictures of the people? Oh, well, for some reason, there's only a person in case study 3. Anyways, you're learning about the different cases and then you're doing certain tests in the case study to try and identify the bacteria and then isolate the bacteria. Tell me what the bacteria is which is causing the patient's infection. Any question about any of that? And I think there's 5 case studies. Yep. All right, if you have any questions, I'll be here until 3:45. I think I'll stay on until 4 because you might have some questions in the case study and you might not get to that until 4 o'clock. All right, any questions? All right, go ahead and start working on the lab. Like I said, this is a longer lab. It'll take you more time to do, but you have a week to get it done. All right. I'm going to use the bathroom, but I'll be back in a few minutes.